Well, good afternoon. It's nice to see so many of you at the first session of the 2024 San Antonio Breast Cancer Symposium. Please don't be shy. This is a big room. Please come on down to the front. The speakers do not like to look at rows of empty chairs at the front and try to see you all the way at the back of the room. I think we have a real exciting program for you this afternoon, mechanisms of obesity-related risks for breast cancer and approaches to risk reduction. I'm going to give a brief introduction for the first few minutes. And remember, this is the obesity session. If you're looking for sex, drugs, and rock and roll, a survivorship session, it's down the hall and around the corner. It's not advancing. I have no relevant financial relationships. I think most of this audience is very familiar with the fact that obesity is associated with an increased risk of postmenopausal breast cancer. It's also associated with an increased risk of mortality from breast cancer, as well as all-cause mortality after a breast cancer diagnosis, especially if women are in the higher obesity range, severe or morbid obesity. And this has really been emphasized in this updated publication from the Women's Health Initiative. What's a little bit disconcerting is there has been a doubling of the obesity rate just in the time that I have been in practice over the last 50 years. And most concerning is there has been a quadrupling of the severe obesity rate in adult women. And this is in part due to the increase in early life weight gain. Well, how in the world has this come about? Well, the average baby boomer had a fairly normal BMI of about 24 kilograms per meter squared when they were in their 20s. Contrast that with the average BMI of the millennials, Gen X, and Z of 27. These people, on the average, are already overweight when they're in their 20s. The weight gain in young women is substantial in this day and age and sets the stage for midlife obesity. The average high school grad has a BMI of 22 kilograms per meter squared, still in the normal range. Then they go to college, and you've all heard of the freshman 15 pounds. It's really more like 3 kilograms, but still most early adults gain weight in college. Then comes marriage or cohabitation, either one, with a 2.5 kilogram weight gain. And it really doesn't make any difference if the woman has healthy lifestyles and low BMI at the time she gets married. If her mate has unhealthy habits, she will tend to adopt his habits over a period of time. The big one, as we all know, is babies. Postpartum weight retention a year after birth averages about 5.4 kilograms or 11 to 12 pounds. What this all results in is a BMI of about 29 when women are 39 and ready to go into midlife. You couple that with a 0.7 kilogram per year weight gain in middle age, and the 45% obesity rate in US women between the ages of 40 and 60 is not surprising. Well, what are the reasons for this excessive weight gain in early adulthood? Probably the biggest one is a drop in physical activity post-adolescence. High school kids going from playing soccer 3 or 4 days a week to, for the most part, sitting in front of their computer, and their idea of exercise is perhaps walking their pets. They leave home, they eat more obesogenic food, And many of them lack cooking and food selection skills because they never learned it at home in the first place. Over a third of US adult women say they don't cook at all. Luckily, the cooking amongst men is starting to increase. But the lack of cooking, busy schedules, and a cheap food means that a lot of people are eating, especially young adults, are eating more fast food. And studies have shown that having as little as 2 fast food meals per week increases the rate of obesity significantly. Well, what causes the high postpartum weight retention, even in excess of the 5 kilograms? Well, high pre-pregnancy weight is associated. African American race, being very young, and being in a low socioeconomic status category. A lot of this can either be attenuated or avoided with prolonged breastfeeding, at least for 6 months, and moderate intensity exercise. You'll see that exercise is a recurrent theme in this session. What about midlife? Well, probably the biggest reasons there are continued low physical activity, and maybe people in midlife don't eat quite as much in the way of double cheeseburgers, but they tend to still have suboptimal diet quality. Too many of those double caramel lattes and bagels for breakfast and a couple glasses of wine for dinner. There's also a pretty much unavoidable age-related decrease in lean mass and metabolic rate. All the stress of jobs and raising the family oftentimes lead to insufficient sleep, and then you get to menopause transition and nobody sleeps. Also, some commonly used medications in midlife, including SSRIs, antihypertensive drugs such as beta blockers and angiotensin receptor inhibitors, and progestins, often used for birth control or even to control perimenopausal bleeding. Obesity also is associated with epigenetic changes. It modifies immune cells, oncogenes, and tumor suppressor genes. And as opposed to increases in insulin and pro-inflammatory adipocytokines and hormones, how readily can we really change these epigenetic changes that can really modify oncogenes and tumor suppressor genes? Catherine Duggan is going to talk to us about this in just a minute. Sustained weight loss of 10% or more, which oftentimes is associated with a visceral fat loss of 20 to 30% or more, likely reduces obesity-related breast cancer risks. Neil Iyengar from Memorial Sloan Kettering is going to go over a number of pathways to obesity reversal. We all know that lifestyle alone is very difficult to achieve and sustain a 10% weight loss, particularly the sustaining part. Bariatric surgery is commonly associated with at least a 20% weight loss and a 30 to 40% reduction in breast cancer risk. But there's only about a 1% uptake in the US of risk-eligible women, and this is predominantly due to side effects of the surgery as well as expense. The incretin analogs, semaglutide and tirzepatide, are associated with a 10 to 20% weight loss in clinical trials, but there's little evidence in early observational studies of change in breast cancer risk. And I think we need to think about why that is so. Our third speaker, Dr. John Jakicic, who's an expert in exercise and physical activity, particularly as it pertains to weight management, is going to talk to us about how exercise acts to help mitigate the adverse metabolic effects of obesity, and importantly, helps prevent regain after weight loss. So with that, I would first like to introduce Dr. Catherine Duggan. Unfortunately, she had some family emergencies and is unable to be with us here in person today. Catherine, again, is an epidemiologist and an expert both in risk factors for breast cancer, particularly as it pertains to obesity and epigenetics. She's going to explain to us what epigenetics are, and then talk to us about how epigenetics could be used to perhaps monitor our interventions as we go forward. And Catherine, I think we are ready for you.
Okay. Thank you so much, Dr. Fabian, and good afternoon, everyone. And thank you for the invitation to speak at this conference. And I'm sorry that I can't be there in person. Next slide, please. I have no financial relationships to disclose. Next slide. So to begin, let's clarify briefly the distinction between genetics and epigenetics. Genetics deals with heritable changes in gene function that are directly tied to alterations in the DNA sequence. In contrast, epigenetics refers to heritable changes in gene expression that occur without any change to the underlying DNA sequence. A key feature of epigenetics is its responsiveness to external factors, with diet and environmental exposures major regulators of these processes. And this enables cells to adapt dynamically to changes in their conditions or local conditions, and can modulate gene expression by either suppressing or enhancing transcriptional activity. What's important to remember is that epigenetic regulation is dynamic and theoretically reversible, which opens interesting possibilities for clinical interventions that target diseases influenced by epigenetic modifications. Next slide, please. So we'll just briefly go over the different mechanisms that drive epigenetic regulation. First of all, DNA methylation involves the addition of methyl groups to cytosine residues, typically at CPG sites in DNA, which are typically located in 3' untranslated regions of genes that are critical for controlling transcription. Hypermethylation, where CPG sites are densely methylated, which are known as CPG islands, are usually associated with transcriptional repression by reducing messenger RNA transcription, and this in turn decreases protein expression. Histones are structural proteins that organize and compact DNA into chromatin, and during gene expression, histones unwind, enabling transcriptional machinery to access specific DNA regions. Post-translational modifications of these proteins, such as methylation, alter the chromatin structure, influencing gene expression. And depending on the modification, transcription can either again be enhanced or repressed. These chemical groups are derived from cellular metabolism, meaning that metabolic changes can directly impact the availability of these modifying molecules and in turn influence histone modification patterns. Finally, non-coding RNAs, including long non-coding RNAs, circular RNAs, and probably the most well-known microRNAs, which are small molecules about 18 to 22 nucleotides long. These bind again to the 3' untranslated regions of target microRNAs, and in all cases, they block transcription and then prevent protein synthesis. Next slide, please. So very briefly, there are 3 categories of proteins involved in regulating these epigenetic processes. Writers that introduce chemical modifications on DNA and histones, readers and erasers that remove these marks. While we won't discuss these in detail today, it's important to note that some of these proteins, especially DNA methyltransferases, are being explored as novel drug targets in various diseases. Next slide, please. This slide shows a hierarchical overview of molecular processes. First of all, we have the genome, and that provides raw genetic material for cellular function. And heritable DNA variation is driven by acquired mutations, rare variants, and single nucleotide polymorphisms, for example. Sitting on top of the genome, the epigenome acts as a dynamic interpreter that translates both internal and external signals into localized transcriptional control. The epigenome governs the transcriptome, which increases or decreases transcriptional activity depending on the signals derived from the epigenome. And finally, on the right, we have the proteome and metabolome, which are the functional endpoints of this molecular cascade. Next slide, please. So an individual's genome, which is represented by the blue figure at the center of the slide, this exists in a complex network of influences, with the epigenome serving as a bridge between environmental signals and cellular adaptations. Sociological determinants of health, such as education, income, and geolocation, can directly or indirectly influence the epigenome. Pollution of the built environment, exercise, diet quantity and quality, and other aspects of the lived experience can dynamically alter cellular processes. For example, energy storage was historically crucial for our ancestors' survival, with epigenetic mechanisms allowing for modulation of lean muscle mass when needed for activities like hunting or conservation of energy in times of scarcity. And today, these same mechanisms interact with very different modern dietary and lifestyle patterns, which influence body composition and metabolic health. So we have the microbiome, which plays a critical role. Dietary and lifestyle factors influence and shape the content of the microbiome, and that in turn exerts local and systemic epigenetic effects in the host. And then, complicating matters further, there's the impact of transgenerational epigenetic effects, where environmental exposures experienced in one generation influence the epigenome of subsequent generations. So all of these factors converge to influence the phenotype, such as the BMI categories that a person has and metabolic health, which vary even within these categories. And this phenotypic and metabolic variation in turn feeds back to alter the epigenome, creating a dynamic and interconnected loop. Next slide, please. So we mentioned in the previous slide that environmental exposures in one generation appear to influence the health and epigenome of subsequent generations. And unfortunately, some of the most compelling epidemiological evidence comes from studies of historical famine. So during the Dutch hunger winter of 1944, pregnant women had access to about 400 to 800 calories a day. So 6 decades later, the children maintained hypomethylation of the IGF-1 gene compared to their unexposed siblings. These individuals had higher rates of hyperglycemia, type 2 diabetes, and obesity, and similar findings observed in the Chinese Great Leap Forward famine of 59 and the Biafran famine of 1968. And worryingly, higher obesity rates persisted into the second generation. While there's strong evidence for transgenerational epigenetic effects in plants, it's less clear in humans. One factor to consider is that during early embryogenesis, most methylation marks are purged to create pluripotent stem cells. But there are potential areas of differentially methylated regions that escape reprogramming that could be a source of genomic imprinting. And there's evidence that RNA, small non-coding RNAs, are transmitted from parents to embryos. Next slide, please. So there's a reciprocal relationship between obesity and DNA methylation, with obesity causing altered DNA methylation, and that in turn predisposes individuals to obesity. These epigenetic changes in turn contribute to breast cancer development with mechanisms such as persistent inflammation associated with obesity, driving epigenetic changes in breast tissue, altering gene expression. An interesting study looked at methylation or his chromatin modifications of the melanocortin 4 receptor gene in the brain of mice, which plays an important role in body weight regulation. Next slide, please. So over the next few slides, we'll discuss the role of epigenetics in prediction and prevention, intervention effects, and emerging therapeutic options based on epigenetics. Next slide, please. So you may have heard about epigenetic clocks. So these are developed using data from high throughput DNA methylation arrays to estimate biological age rather than, as opposed to chronological age. So they rely on machine learning tools that incorporate data from both the methylation pattern and phenotypic data such as waist to hip ratio, complete blood counts, blood pressure, for example. The first generation clocks were developed in 2013, and we're now on third generation clocks, which are like the Dunedin PACE, which was developed in 2020, which uses data from just 46 sites, methylation sites, and is more accurate. So why is this important? Well, researchers have found that by comparing chronological age, so the actual age of an individual, with the epigenetic age, clock age, can provide insights into aging and disease. So the epigenetic age acceleration is when biological age outpaces chronological age. So if somebody is 45 and their epigenetic age acceleration value is 65, they're more prone to conditions such as frailty, increased cancer risk, and so on. Next slide, please. So epigenetic age acceleration is associated with increased breast cancer risk, with several case control studies using different clocks showing increased age acceleration in cancer cases compared to controls, suggesting it might be useful as a way to stratify breast cancer risk. Next slide, please. So in breast cancer, it's well known that the loss of gene expression plays a critical role in cancer progression, such as P53 is a well-known tumor suppressor. What is interesting, though, is the loss of expression is 10 times more likely to be caused by, more frequent rather, by hypermethylation of promoter CPG islands versus accrued mutations. And in breast cancer cells, this loss of function is particularly significant, with several master epigenetic regulators identified as readers, writers, and erasers that we mentioned earlier that contribute to these changes. Next slide, please. So cells within a field defect, that is regions that are predisposed to cancer, show an increased frequency of epigenetic alterations. One example is DNA methylation changes observed in histologically normal breast tissue adjacent to breast cancers. And in a case-controlled study, methylation patterns predicted whether or not cells were cancerous in adjacent assumed normal tissue and healthy control tissue. And this suggests that epigenetic profiling of tissues might provide indicators of cancer risk or more aggressive cancer types, depending on the methylation patterns. Next slide, please. So over the next few slides, we'll explore how interventions influence the epigenome in individuals with obesity. So from we know obesity accelerates epigenetic aging, and this effect does not appear to be reversible in the short term, even after rapid weight loss through bariatric surgery. Metabolites are critical for epigenetic regulation. If you remember, they need acetyl groups or methyl groups. And these in turn are influenced by obesity-associated dietary patterns. These patterns shape the metabolome, and that in turn affects DNA methylation. So a key question is whether weight loss or exercise can reprogram epigenetic marks to reduce cancer risk and reverse aberrant methylation patterns. And while evidence suggests that some of them can be reversed, the extent of programming required to mitigate cancer risk remains unclear. Next slide, please. So an interesting study, it was 2-year called the Caloric randomized control trial, randomized 220 normal weight adults to either the control ad libitum diet or 25% calorie reduction. And they found that calorie restrictions slowed the pace of biological aging, but there was no reversal of existing patterns. And the sister study assessed eating patterns and found that healthy eating indices were inversely associated with epigenetic age acceleration, meaning that better diet quality slowed epigenetic aging. Next slide, please. So as discussed earlier, microRNAs bind to regulatory regions of messenger RNA and block transcription. And there's dysregulation of microRNAs in both obesity and breast cancer. And we also know that evidence suggests that weight loss can normalize levels of microRNA. So we selected 23 microRNA targets that are involved in the regulation of pathways associated with sex steroid hormone pathways, breast cancer risk, adipogenesis, inflammation, and angiogenesis. And we measured them at the beginning and follow-up in participants of the Nutrition and Exercise for Women study. Next slide, please. So the new study randomized 439 women with a BMI of over 25 who were postmenopausal, healthy, and sedentary. They're randomized to either a dietary weight loss intervention with the goal of 10% weight loss by 3 months and maintenance thereafter, an exercise intervention, combined arm, and control. And the primary aims were to test individual and intervention effects on sex steroid hormones, insulin resistance markers, and inflammatory markers. There's a variety of secondary aims, including assessment of intervention effects on microRNA that I've just mentioned, and line 1, which is a marker of global methylation patterns. Next slide, please. So we found a statistically significant decrease in microRNA in participants who lost weight, with the greatest reductions in participants who lost more than 10% of their baseline weight. So microRNA 122, what does it do? It's reduced, it's elevated in individuals with metabolic syndrome and type 2 diabetes. And in experimental models, a decrease in circulating 122 modifies glucose uptake in cells, and in animals decreases metastatic potential. We found no effects on other microRNAs, and there was no change in global methylation from baseline in 12 months. Next slide. An intriguing study published this year showed that adipose tissue retains epigenetic memory of obesity even after weight loss. And unfortunately, adipocytes were primed to revert to a new obesogenic state post-intervention after caloric restriction ends. Next slide. So finally, a note about the role of epigenetics in therapeutics. Selective estrogen receptor modulators that bind to estrogen receptors have shown epigenetic activity in breast cancer. So they increase production of a protein called TET3 that demethylates, so removes methylation from CpG sites and reactivates silenced tumor suppressor genes. Drugs like azacitidine, I can't pronounce that, already in use for hematological malignancies and bladder cancers, act at low doses to reactivate silenced tumor suppressor genes, again, by removing methylation. Finally, a fascinating study was just published this year where researchers using CRISPR technology were able to edit the epigenome by adding or removing CpG marks in target promoters. And they tested this in mice and removed, were able to silence the expression of a gene involved in LDL cholesterol clearance that was durable up to 1 year. So because this doesn't alter primary DNA sequences, it's theoretically reversible, which is a very exciting potential to develop targeted and reversible epigenetic interventions in cancer prevention and therapy. Next slide, please. So to finish up, an overview of some of the future directions for epigenetics. DNA methylation patterns hold promise as predictive markers for breast cancer risk. Some of these are already in the clinic for advanced, predicting the trajectory of advanced cancers, although it's not yet FDA approved. For patients with obesity, tracking changes in DNA methylation patterns could offer a way to monitor shifts in breast cancer risk. And these could also inform personalized treatment plans, tailoring interventions based on an individual's unique epigenetic profile. Emerging new generations of epigenetic clocks with increased predictive accuracy will provide new insights into biological aging and cancer risk, and potentially developing breast cancer specific epigenetic clocks. And lastly, future epigenetic therapies targeting metabolic pathways might offer a novel approach to prevention and treatment. Next slide, please. I'd like to thank my colleagues at the Fred Hutch and gratefully acknowledge BCRF funding. Next slide. And thank you for your attention.
That was an extremely informative and fantastic talk. For the most part, we're going to take questions during the panel discussions, and you can either come to the microphone during the panel discussion or access the QR code and ask your questions that way. However, we're running about 3 minutes ahead of time. So if there are any specific questions for Dr. Duggan at this time, we can go ahead and entertain some. Catherine, I was kind of dismayed that even if we do reverse obesity and adopt a healthy diet and exercise like crazy, we are not going to reverse epigenetic aging. We are just going to tone it down a bit. Is that right?
Yeah, unfortunately, that seems to be that seems to be the case. And not only that, but fat cells also seem to maintain a transcriptional pattern even after weight loss that resembles the obesogenic state. So this really suggests that early life events or interventions to decrease obesity would be very valuable. That, preventing obesity rather than intervening at a later date will have the most impact on metabolic health.
Okay, so I think the point is we probably need to have more cultural and legislative modifications to prevent people from becoming obese in the first place.
Yeah, absolutely. And education about, that while the phenotype might change metabolically, while there will obviously be improvements, some of the I don't like to use the word damage, but I suppose some of the effects are already embedded at that point.
Okay.
But certainly there's so many environmental pressures that are not in our metabolism's best interest. So I think there's a lot of work to be done, both at policy levels and education. Thank
you very much. We have one quick question at the microphone, and then we're going to have to move on.
Hi, my name's Mark Tuttle. I work in Oxford in the UK. Thank you very much for an interesting talk. Oncologists like scores and to help guide their therapies, and you mentioned about this epigenetic aging score. Is it something that you can order this? Is it accessible? Something we could use to help direct interventions?
So the most recent third generation one is it's a computational method. So you punch in once you have the data from your patients, you select you run those data through, and then you input information such as waist tip ratio, blood pressure, in some of them, oddly enough, educational attainment, and it gives you the epigenetic age. So it gives you somebody is 40 and it tells you their age is 60, then clearly they're at risk for a number of different, chronic health issues. There are a number of not yet FDA approved methylation that analyze methylation patterns, and they're actually in use for patients with advanced cancer. So they run the test and give you back a score, and depending on the pattern, it can impact what kind of therapies they have. So it's still in its infancy. It's computationally quite intense, but things are going to improve. So I suspect that in a few years there will be scores based on this.
Thank you. Is it possible to name of the score?
Yeah.
The name of the scores, the name of the score we could calculate?
It's called the Dunedin PACE epigenetic clock. So
thank you.
Okay, we really have to move on. And perhaps this score is not quite ready for prime time, and perhaps that's more information than some of us want to know. So our next talk is Achieving Sufficient Fat Loss and Metabolic Improvement with Incretin Mimetics to Reduce Cancer Risks. Dr. Neil Iyengar, and he is associate professor at Memorial Sloan Kettering. He's a breast oncologist and leads the MSK Healthy Living program there. Neil?
Thank you, Dr. Fabian. Good afternoon, everyone. So I'm going to shift gears a little bit and speak about an intervention, but first I'm going to lay some groundwork and describe some of the mechanistic work that our group and others have conducted to look at or investigate the impact of adiposity on tumorigenesis and cancer progression. I'll then move into the use of incretin mimetics and how we may or may not be able to utilize these tools to reverse some of that pathophysiology. I'll just comment, we did receive quite a number of questions just now, some of which I think I'll actually hopefully address in this talk, and then if not, we'll get to those in the panel discussion. With that being said, here are my disclosures. None of these relationships are related to the topic of interest today. I'll start with an overview mechanistic slide that we actually published back in 2015, and so there are certainly some updates that need to be included, but overall I think this helps to encapsulate or summarize both the systemic and local impact of adiposity on cancer growth pathways or potential cancer growth pathways. I think the systemic pathways are well recognized. Certainly we know that excess adiposity is associated with increased amounts of circulating free fatty acids, hyperinsulinemia, hyperglycemia, increased bioavailable estrogens in the circulation. At the local level, we also see evidence of inflammation, particularly adipose tissue inflammation, which gives rise to a microenvironment that is enriched with tumor growth factors. In addition to this, we see a pro-estrogenic microenvironment, which I'll get to in subsequent slides, and this really establishes both a local and systemic environment that's prone both for tumor growth as well as propelling the growth of established tumors. So here I'll comment a little bit more on the impact of adiposity on the microenvironment. And of course, this is relevant as we start to think about how weight loss interventions, and particularly interventions that significantly induce significant weight loss, such as the Incretin mimetics, bariatric surgery, may impact these effects of adiposity. So as we all know, the breast is largely comprised of adipose tissue, and the tumor often directly interfaces with that adipose tissue if it's present. Now, certainly we're talking about prevention here, but I think we can apply some of the learnings in the post-diagnosis setting to what might occur in the pre-diagnosis setting or even at the tumor initiation setting. Our group and others have described the presence of inflammation, adipose tissue inflammation, in the setting of obesity. This is histologically recognizable by the presence of a crown-like structure, which is essentially a dysfunctional adipocyte that is surrounded by activated macrophages that take on a metabolic phenotype in an attempt to clear that dysfunctional adipocyte. This is an inefficient process that does not lead to clearing of that adipocyte, but rather creates a focus or a hotspot of inflammation. I mentioned that this is histologically recognizable, or in other words, a histologic biomarker. On top here, you see a CD68 immunostain that's highlighting those macrophages surrounding that adipocyte. On the bottom is a classical H&E stain from a patient who underwent mastectomy and donated her fat tissue. Now, through a series of bench-to-bedside experiments led by Dr. Andy Dannenberg and Christy Brown, we were able to recognize that the presence of adipose tissue inflammation not only leads to that pro-inflammatory microenvironment. Many of those factors are direct tumor growth factors, those cytokines that you see listed on the slide, but we also know that many of those cytokines can directly induce CYP19, which of course is the gene responsible for encoding aromatase, or the rate-limiting enzyme in estrogen biosynthesis, thereby generating a pro-estrogenic environment on top of that pro-inflammatory microenvironment. This has implications both for hormone receptor positive breast cancer as well as triple negative breast cancer, given the presence of those inflammatory foci or inflammatory cytokines. Now, when we look at the prevalence of adipose tissue inflammation stratified by the standard tool of body mass index, we do see that the majority of individuals with classical obesity or a BMI of 30 or greater do have adipose tissue inflammation evidenced by this histologic biomarker. We also see that the majority of individuals, though a lower proportion that are overweight, also have adipose tissue inflammation in the breast. And intriguingly, and I'll come back to this, there is a population of normal weight individuals with a BMI less than 25 who also harbor adipose tissue inflammation. When we look again in the post-diagnosis setting, we see that the presence of adipose tissue inflammation or these crown-like structures in the breast are a prognostic biomarker and are associated with an increased risk of distant recurrence-free survival. Here I'm showing you the Kaplan-Meier curve for the multivariate model that controls for nearly every known prognostic factor in breast cancer, with the orange curve highlighting those individuals with adipose tissue inflammation. Now, in subsequent work, other groups have validated these findings in independent cohorts, and we've also seen data presented from several groups indicating that in the pre-diagnosis setting, the presence of adipose tissue inflammation or these crown-like structures are associated with an increased risk of breast cancer. In this particular study, this was in the setting of pre-existing benign breast disease. So now let's take a look at that group of individuals that I mentioned earlier who have a normal body mass index and adipose tissue inflammation. And I highlight this group because, as I mentioned at the onset, I will be speaking about incretin mimetics, and we know that these drugs can be dosed to induce varying levels of weight loss. And indeed, the first indication for incretin mimetics were in the setting of diabetes and did induce weight loss, but to a lesser degree than what we see at higher doses or at doses indicated specifically for weight loss independent of diabetes. So this raises the intriguing possibility that if we are addressing an individual who may have metabolic obesity in the setting of normal weight, perhaps we can utilize this pharmacologic approach in a way to induce recomposition of adiposity to lean mass ratio. So in work in collaboration with Dr. Christy Brown, we demonstrated that even in normal weight individuals, the presence of adipose tissue inflammation or those crown-like structures are associated with an increased expression of aromatase in the breast tissue, and this is by aromatase immunofluorescence. Here you can see two representative images. The individual on the left is a breast section taken from an individual without adipose tissue inflammation, while the individual on the right does have evidence of adipose tissue inflammation. The red is highlighting or immunofluorescing aromatase. There does appear to be a dose-response relationship between the level of inflammation and the degree of aromatase expression. When we look at the size of the adipocytes, we also see an association between adipocyte diameter and adipose tissue inflammation. As you might expect, the larger the adipocyte, the greater the degree of inflammation. That's the crown-like structures per centimeter squared of adipose tissue. And similarly, we see an association between larger adipocytes and increased aromatase expression in the breast tissue as well, noting again that these are in normal weight individuals. The size of the adipocyte is a surrogate of body composition or total adiposity, so this was our first clue that these individuals with adipose inflammation and a normal body mass index are likely to have increased levels of total body fat or excess adiposity. Now, that's demonstrated over here on the right side. This is a study where we conducted DEXA scans to measure body composition in individuals who are undergoing mastectomy, and there you can see color-coded in the blue are those individuals who do not have adipose tissue inflammation, and in the maroon are those individuals with adipose tissue inflammation. And in this normal weight group, those folks with adipose tissue inflammation had higher levels of total body fat percentage, and particularly trunk fat mass shown on the bottom there. Now, when we take a look at how this might translate to breast cancer risk, this was a study that we conducted in collaboration with the Women's Health Initiative. And for the sake of time, I'll go straight to the punchline here with the forest plot. And essentially what you can see here is that the higher the level of body fat, the higher risk of developing invasive breast cancer after menopause. So this is postmenopausal breast cancer. This is all subtypes, both hormone receptor positive and hormone receptor negative. And essentially, you can see those individuals in the highest quartiles of whole body fat as well as trunk fat mass with a slightly larger effect size had nearly a doubling or slightly over a doubling in their risk of developing invasive breast cancer, despite having a normal weight. I should emphasize that here, that we focused specifically on those individuals in the Women's Health Initiative who had a normal body mass index. Now, for the purpose of metrics, if you're wondering what the cutoff is in terms of whole body fat percentage, we saw that the risk of invasive breast cancer went up after a whole body fat percentage of about 33%. And that is consistent with the WHO definition of metabolic obesity in the setting of normal weight. Now, in further investigation and study led by Priya Bhardwaj and Christy Brown's lab, we focused particularly on individuals who have a genetic predisposition to developing breast cancer. So this was a cohort of individuals that we had enrolled at MSK who were undergoing prophylactic mastectomy due to the presence of a germline BRCA1 or 2 mutation. And we looked at tissue measures of epithelial DNA damage, specifically gamma H2AX immunohistochemistry, which is demonstrated here both in terms of the H&E stain, and we also see immunofluorescence staining. I'll show you here the curve or the relationship with body mass index, and we see that in the presence of a germline pathogenic BRCA mutation, the higher the BMI, the increased gamma H2AX foci, or increased evidence of DNA damage. And this is consistent with some early epidemiologic data which suggested that the penetrance of breast cancers is indeed higher when obesity is present in the setting of a germline BRCA mutation. So this also raises the possibility that we should be including those individuals with genetic syndromes and obesity in our prevention studies that are focused on metabolic interventions. When we look at circulating metabolic markers such as insulin, glucose, and the homeostatic model for insulin resistance, we see an association between those circulating metabolic markers and increased epithelial DNA damage in the breast tissue. The immune microenvironment is also dramatically transformed in the setting of excess adiposity or obesity, and this is important given emerging evidence that weight loss and possibly the incretin mimetics may also impact the immune microenvironment. So I'll just cut to the chase here. This was a computational method where we looked at gene expression signatures of immune cells, and the bottom line here is you essentially see a suppression of the local immune system and immune cell infiltration, particularly cytotoxic T cells, natural killer cells, as well as their cytotoxic function in the setting of excess adiposity and obesity, despite the presence of inflammation. And this has been recapitulated in other studies as well, raising the question of, are tumor cells able to evade our immune system defenses in the setting of obesity, and can we reverse that with weight loss? And so that, of course, is the hypothesis that all of that pathophysiology that I just showed you summarized in this schematic here may be reversible with weight loss, and many groups have looked at lifestyle interventions, which I'll show you some of that data for the purposes of weight loss, but particularly for the purposes of inducing biologic changes that then reduce the risk of developing breast cancer. And we heard from Dr. Duggan, and also based on Dr. Fabian and Dr. Hursting's work, that there does appear to be a threshold of weight loss that is optimal for reversing some of those biologic changes, about 10% weight loss or greater in postmenopausal women. And I'll show you some observational data that corroborates that evidence. So these data were provided by Dr. Hursting, and here we see that caloric restriction in preclinical models can reverse some of that obesity-driven suppression of immune suppression, or suppression of immune hallmarks, rather, in the microenvironment. And this is a preclinical model here. I won't go through all of this for the sake of time here, but essentially a lot of what we saw in the human tissues in the setting of obesity can be, or has been shown to be, reversible in preclinical models with caloric restriction. When we look at human studies, I'm going to show you a summary of human studies and some of the seminal trials that have looked at weight loss, starting with the Women's Health Initiative. We also see evidence that those findings with regard to reversal of the biologic or pathophysiology do translate to a reduced incidence or a lower risk of developing subsequent breast cancer or obesity-associated cancer. So this first data set here I'm showing you is from the Women's Health Initiative with a focus particularly on the prospective observational cohort. So over 50,000 women were included in this analysis, postmenopausal women, with a median follow-up time of 12 years. And the finding was essentially that those women who reported a 5% or greater weight loss on average were noted to have a 12% reduction in the development of obesity-associated cancers in general. And there you can see the spline curve with a hazard ratio of 0.88. In the look-ahead randomized control trial, which tested an intensive lifestyle weight loss intervention in overweight and obese patients with type 2 diabetes. The investigators conducted a post-hoc analysis to look particularly at the impact on obesity-associated cancers. And this analysis included over 4,000 women with a median follow-up time of 11 years. The investigators reported a 16% reduction in obesity-associated cancers, particularly in the intensive lifestyle intervention arm, and that's the dark maroon line in the curves there, with the blue line representing the control arm. They also noted a reduction in cancer-specific mortality in those individuals who were diagnosed with an obesity-associated cancer. And I do think that that's important data as well. Oftentimes we're asked the question of, well, I lead a healthy lifestyle, I lost weight, I was still diagnosed with cancer. And we have evidence from this and several other trials indicating that that weight loss prior to the diagnosis of cancer and subsequently thereafter may still improve outcomes in terms of cancer-specific mortality. These data now show more intensive weight loss through bariatric surgery. This is the Splendid matched cohort study, which matched patients in a 1-to-5 manner, those patients who had received bariatric surgery procedures versus those patients who did not receive surgery. And with 6.1 years of median follow-up time, a 32% reduction in obesity-associated cancers was noted. And I apologize, the curves are so small here, but what I want to point out here is that there does appear to be a dose-response effect in terms of the degree of weight loss and the incidence of obesity-associated cancers. So we see in those individuals with the highest amounts of weight loss, this is divided up by quartiles here, so we're talking over 30% weight loss here. Those individuals had the lowest incidence of obesity-associated cancers. So now what about pharmacotherapies? I showed you observational evidence and lifestyle interventions, and as we all know here, there's a rich history of using non-tumor directed approaches for cancer prevention and even testing them in the therapeutic setting. I put up this slide not to go through each and every one of these approaches, but to essentially emphasize the point that many agents have been hypothesized to have a cancer protective effect. And if you're interested in a very comprehensive summary of all the potential agents that have a potential cancer protective effect, and some of which are in testing right now, I direct you to this review article here. Let's now focus specifically on incretin mimetics. Of course, these are the sensational weight loss drugs, if you will, that have garnered a lot of attention because of the degree of weight loss that they induce. And I just want to spend a minute or two walking you through some of the mechanisms, because these mechanisms are and might be relevant to cancer growth or cancer risk reduction here. So in this schematic here in panel A, you can see the insulinotropic effects of GLP-1 and GIP receptor agonism. And essentially, these drugs work by increasing insulin secretion. Now, I will say it is more complex than that because it depends on the basal glycemic state of the host, whereby in the setting of, as I'll show you on subsequent slides, you don't actually induce hypoglycemia in a normal glycemic state. So there are regulatory and feedback mechanisms at play here. But this schematic is specifically isolated to the pancreatic beta cell here. And one of the things that I think is important to point out here is in panel B, you see the non-insulinotropic effects here, and you actually see mediated through EGFR an activation of the PI3 kinase mTOR pathway, which of course in a beta cell, in a pancreatic beta cell, is an effect that we want, pro-proliferative anti-apoptosis, and thereby protecting pancreatic function and insulin homeostasis. But that does raise the question, what is the impact on this signaling transduction pathway in cancer cells or in epithelial cells that might be transitioning to a carcinogenic state? The other question is, do all of the other effects of incretin mimetics, particularly adiposity reduction, as I'll show you on subsequent slides, overcome a potentially pro-cancerogenic effect at the cellular level? And does this effect persist, particularly in cancer cells? These are all questions that are currently under investigation. The schematic at the bottom is essentially showing you, and I'll show you a bigger one in the subsequent slide here, that these agents have multiple effects. Now, we have single agonists that target the GLP-1 receptor. We have dual agonists that target the GLP-1 and the GIP receptor, and then we have, of course, have emerging triple agonists that also target the glucagon receptor here. And in this slide, I think, let me come back to the panel on the left there, but what I want to show you, if I can pull it up here, there it is color-coded by receptor agonism here. You can see that there are really quite a number of effects, both locally, as I showed you on the previous slide, as well as systemically through dual and triple receptor agonism here. Most of, if not all of these effects are pro-metabolic, essentially protecting adipose tissue function, decreasing or normalizing hyperglycemia, maintaining glycemic homeostasis, as well as protecting skeletal muscle function. We do know that these drugs also slow gastric emptying, which may be a mechanism through which they induce energy restriction. Relevant to the breast cancer space, many of these agents have also shown positive effects on bone metabolism, bone protection. And we also have emerging evidence that there are CNS effects potentially that not only regulate hunger and our food intake, but may also regulate memory and even addiction pathways. Now, on the left is a schematic that I was alluding to earlier, whereby the incretin effect is not a simple increase of insulin secretion, but a feedback mechanism which regulates insulin homeostasis such that the underlying glycemic state of the host impacts what the ultimate effect will be on blood sugar levels. And essentially what you can see here is that in the normal glycemic state, you essentially get a regulation or maintenance of that glycemic state, whereas in the hyperglycemic state, these drugs are incredibly effective at lowering hyperglycemia and blood sugar levels. And I think in clinical practice, the positive ramification of this, of course, is that they don't induce hypoglycemia, and you don't see the effect that you might see with insulin, for example, which can be challenging to manage with a yo-yoing effect if you're using too much versus not enough insulin. Now, I want to show you some observational evidence that was recently published by Professor Berger's group here, and I also want to take a moment to acknowledge his incredible impact on the field and many of our careers. So we saw data that was recently published by his group where national medical records were examined, and we're talking millions of patients here, over 15 million patients who were prescribed incretin mimetics, predominantly single and dual receptor agonists, for the treatment of type 2 diabetes. And with a median follow-up time of about 15 years here, you can see the impact on several obesity-associated cancers. In fact, the incidence of most obesity-associated cancers were reduced with the use of these drugs. Now, there are several open questions here. The dosing of the drug matters, combinations with other anti-hyperglycemic medications for the treatment of type 2 diabetes and the impact there. And there was a lot of modeling that was done to try to isolate the incretin mimetic effect here. But notably, you can see second on the list there is breast cancer. And we don't see a statistically significant protective effect on breast cancer. And again, they also focus particularly on postmenopausal breast cancer, and a protective effect was not reported. However, I will note that this is discrepant with the biologic hypothesis, as I've showed you on prior slides. And my thoughts here, and several of us are digesting this data, I will say. And the follow-up time here is fairly limited for a breast cancer prevention study. So I think we still have a lot of open-ended questions here. I mentioned dosing, I mentioned combination drugs, but I think particularly the driver here is the follow-up time, but also the window of intervention and when adiposity is most impactful on breast cancer risk. I want to show you some preclinical evidence that's been reported preliminarily by Dr. Hursting's group here, which supports an anti-cancer effect of dual receptor agonism. Now, this is not particularly a reduction in tumor volume, but rather a prevention of further growth here. So this is an orthotopic model done on a preclinical obese background here, induced by diet-induced obesity. This is a triple-negative breast cancer model, EO771, treated with tirzepatide, a dual receptor agonist here. And I'm running a bit low on time here, so I'll just cut to the punchline here, and that is essentially what you see here is that in the setting of tirzepatide, smaller tumor mass, but notably when we look at caloric restriction, you also see a similar, if not more, pronounced effect. And sorry, this is so blurry here, but what this is showing you are circulating metabolic markers, which are significantly improved both by tirzepatide as well as caloric restriction. We've seen similar data. This is a colon cancer model, MC38 model, preliminarily published by Dr. Pollack's group. And what I think is particularly interesting in this study here is they also included a preclinical group or model that was pair-fed or calorically restricted to the same degree of caloric intake as the mice treated with tirzepatide. And what you see there in the curves regarding body weight here is that the mice treated with tirzepatide lost the greatest degree of weight here, but when you look at tumor volume, the pair-fed mice, the calorie-restricted mice to the same degree as the caloric intake of the tirzepatide-treated mice had a very similar impact on tumor volume reduction here. And this gets back to Dr. Fabian's comment about lifestyle interventions and caloric restriction being effective, but perhaps difficult to maintain. In the post-diagnosis setting, our group has reported recently at ASCO in a small cohort of 75 women that the use of GLP-1 agonists appears to be safe. We did not note any increase in toxicity, particularly in combination with endocrine therapy. But the mean relative weight change in this small cohort was only a reduction of 5%, suggesting that perhaps there are interactions with cancer therapy or there's something different about the post-diagnosis setting. And we'll see more data that will be presented on Thursday from Dr. Sukumar et al., who looked at a much larger cohort, over 1,000 patients that were treated at MD Anderson. So I'll just put a plug here to come to the poster spotlight discussion on Thursday at 7 a.m., bright and early, where I will be reviewing that data. So I'll end here with some conclusions and some questions, quite a few questions, in fact. So as you've seen, not just from this, but all of our talks so far, is that weight loss, particularly 10% or greater, appears to attenuate the cancer-promoting effects of obesity. We have promising observational data which indicate that the incretin mimetics, both the single and dual receptor agonists, and perhaps more so the dual receptor agonists, may reduce the risk of most obesity-associated cancers, but we do need longer follow-up time, studies with longer follow-up time, to really interrogate the postmenopausal breast cancer question. It appears so far, based on the available evidence, that the effects of incretin mimetics are likely mediated through energy restriction. I forgot to mention in the preclinical studies that in vitro modeling, there was no anti-proliferative effects on the cancer cell lines, but there are ongoing and emerging studies that will help to further address whether or not these agents have direct anti-cancer efficacy. I do think the impact on the fat tissue microenvironment is likely to play a larger role there. And then the impact of incretin mimetics are likely, as I mentioned, to be different in the pre- and post-cancer diagnosis setting. This has implications for the design of prospective clinical trials where we might want to be testing different dosing or duration of these incretin mimetics based on the particular setting that we're testing them in. The key questions that I think are really important to address here, and there are more, these are not all of them, but what is the optimal window for a risk reduction effect to intervene, and what's the optimal duration of therapy with the incretin mimetics to effectively reduce cancer risk? And then the other major issue, as Dr. Fabian alluded to, is what is the impact of weight gain on cancer risk after treatment with the incretin mimetics has stopped, or do we need to continue patients on the incretin mimetics forever? gives rise to the question underneath that, and that is that can we combine pharmacotherapy with lifestyle interventions? And I think this is the most attractive approach as perhaps an induction and maintenance approach, if you will, for inducing weight loss and maintaining that weight loss. And then finally, this gives us a great opportunity to validate metabolic markers as surrogates for cancer risk. Here are my acknowledgments, and I thank you for your attention.
We are running a little late, so there's lots of questions that have continued to come in for both the initial two speakers, but we're going to hold those until the end. Our final speaker is Dr. John Jakicic. He is a professor of medicine at the University of Kansas. He is an incredible expert in exercise and general physical activity, particularly as it pertains to weight loss and weight management. He's had a many year, couple of decades leadership experience in the American College of Sports Medicine. He currently is the vice president of that organization, and he's the first author of the American College of Sports Medicine update on exactly what you're supposed to do for physical activity in 2024. John?
Thanks, Carol. I appreciate the invite to be here today. So just jump right to this. Just one disclosure that really has not much to do with what I want to talk about today. So I like to position physical activity and exercise maybe a little differently than the way you think about it or the way you've heard it presented before. And I like to start by presenting a picture of this young man who in October of 2023 set the world record in the marathon at 2 hours and 35 seconds. And unfortunately, lost his life in a tragic accident in his home country of Kenya soon after that. But I like to think about this, and my guess is that how many of you in the room feel like you could run a 2 hour and 35 second marathon? Okay. So, well, maybe one of the reasons that you can't do that is you can't train the way this young man was able to train. None of us have the time to do that. Go out there and train. You have the trainers around you. How many of you could run it if you had the trainers and the training time that he had? Yeah, probably not many, if any. But also he has this environment around him that allows him to do this as well. And so you start to think and say, well, if I could train and I had the environment, I had the nutrition people around me, I had the right environment, I had the right facilities, I had all these things, could I do it? Well, if you could do it, you wouldn't be sitting here today. And the reason that you can't do it, even under the best of circumstances, is that behind that training and behind that behavior and behind that environment is a biology and a physiology that's going to influence how well you train, how well you adapt to that training. And so as we start to think about the effect of exercise on obesity and its related comorbidities, we really need to be very careful about thinking one size fits all. And we've written guideline papers, position papers. One size when it comes to exercise does not fit all. Just like in medicine, when you treat a patient, one size does not fit all. And too many times I think we get ourselves in this exercise, physical activity box. And I hope I get you out of that box today to think a little bit differently about how exercise might need to be thought about. And so when we think about the complexity of obesity for so many years, particularly in the behavioral sciences, we think about eating behavior, we think about physical activity, and we say if we modify those two things, we're going to get an effect. But when we do that, in the literature, it's clear we get varying effects. The effects are not the same for every person. And why is that? And this is not comprehensive. But these things interact with one another. And particularly I'm interested in the activity and exercise space. And we have found now in numerous studies that when you have somebody begin an exercise program, some of those individuals have a satiation effect to that exercise, and some of those individuals have a hunger effect to that exercise. It's not the same for every person. Some of them get that at a high dose, some of them get it at a low dose. Some of them get it at a high intensity, some of them get it at a moderate or lower intensity. And what we have to remember is as we modify these things, they all filter back on one another. And so as we think about physical activity and exercise, it's not as simple as go out, exercise, get this effect, you're going to burn more calories and it's going to have all the effects you expect it to have. It's not the way that it works, just like it doesn't work that way with other factors of medicine. So as I start to think about the obesity issues and how we treat them and where activity fits, we have these, basically I'll call them 3 pillars of treatment. We have basically lifestyle factors that we can treat. We have bariatric surgery options, and we have pharmacotherapy options. Those are our 3 options that we have. And these things filter on one another. It's not a static thing. And I think the point of this slide is at the heart of it all, even when we have medical treatments for obesity, lifestyle is still important. Lifestyle is still important across the board, and activity and physical and dietary intake and behavior are all critical to that, to make them work. And so as we think about where activity fits, I want you all to think about that it's part of a pillar of treatment, not an independent factor in treatment. It's part of a comprehensive care model. And so as I think about this, I say, well, then what should be our focus when we treat obesity and excess adiposity? In our most recent position paper, we term it excess adiposity, not just excess weight, but excess adiposity. And should that effect be on maximizing weight loss? That's currently what our more current medical treatments are really focused in on. Should it be on achieving a meaningful clinical weight loss that's sustainable? Or should it be on prevention of excess gain, weight gain? I actually think the focus should be on none of them. I think we're missing the mark. I actually think that the focus needs to shift, and I think some of our prior speakers have hinted at this, that it needs to speak to adiposity, adiposity loss, meaningful adiposity loss, and prevention of adiposity gain. That is not one and the same with weight. It means that we have to shift our thinking around this just a little bit. And so we have to take it from a weight-focused approach to an adiposity-focused approach, in my opinion. And I think we have to take it one step further from an adiposity-focused approach to a health-focused approach. Instead of focusing on what happens on that scale, focus on what's not only happening on that scale, but what's happening to the composition of what's happening on that scale and its effect on downstream health indicators. And when we think about this, I think that the focus also has to be on what is the physiology and the biology that's changing behind this that's going to enhance health? And how do we do that? And I think, in my opinion, we have plenty of studies that show that weight loss, adiposity loss, helps with a variety of things. And I don't have enough time today to show you the data that show you that if you just focus on adiposity loss or weight loss, you are not completely enhancing the health of the individual. And I think at the heart of that is activity is critically important to that. And we have shown in study after study after study after study that even though activity and exercise does not add a lot more to the weight loss that you get with diet-induced interventions, things that change eating behavior, the health benefits are magnified dramatically when activity is part of that plan, beyond what you would get with weight. And so from a clinical perspective, if we're really focused on enhancing the health and well-being of a patient, we shouldn't stop at weight loss. We should be stopping at health-enhancing effects of our treatments. And that goes back to these three things. All three of these need to be considered. And most recently, the medications that we're talking about, the pharmacotherapy agents, are really focused in on trying to impact the behavior of eating and the biology that helps to control that in many ways. That's where our focus has been. And this is just a representation of these anti-obesity medications and the trajectory of what we've seen since really this most current evolution has occurred over the last 10 years or so. And you can see they're getting better and better and better. And it was hinted at, we now have triple agents, we have additional, I forget how many is in the pipeline, hundreds are in the pipeline, right? We don't have an answer yet to a lot of these things. These are not going away, and they're going to get better and better and better. So, but what do we know about these contemporary treatments? And this slide needs updated on a daily basis, so I admit that it may be a little dated at this point. But a lot of good things happen, and this is a small fraction of all the good things that happen in addition to the cancer risks and all the other things that are happening. But the one thing that we're seeing is that in addition to these benefits, is that with these medications, we're seeing actually a really large reduction in lean body mass. And a lot of people are getting concerned about that. Even the pharmaceutical industry is a bit concerned about it, and they're trying to create agents that are going to minimize that effect. But I want to make sure that we understand something that's critically important here, and it means we have to understand body composition. Lean mass is not muscle mass. Lean mass is muscle, may be considered bone depending on how you measure it, organ tissue, connective tissue, all kinds of things. Muscle mass is muscle mass. And to my knowledge, there have been no good, big studies, maybe the pharma companies have the data, that have reported on what's happening to muscle mass in these studies of these agents. And only a few have reported on body composition in general, losing good methodology. So as we think about where I go from here and the effect of exercise and physical activity, I want you to remember two things. One is lean mass is not muscle mass, and we should not be using them interchangeably. The second thing is I think that our focus on volume of tissue, particularly lean tissue, maybe muscle, is misguided. And we should be focusing on the quality of those tissues, not the volume. Because after doing probably 10,000 DEXA scans on people in my life and my studies, I can tell you patients with obesity have more lean mass than their less overweight counterparts. It's not an issue of mass, volume. But I can also tell you from a variety of ways of looking at it, the quality of their lean tissue is less good, if you want to put it that way, than their leaner counterparts. And I think that as we think about activity and exercise, we need to think about its effects on the quality of the lean tissue, not the volume of the lean tissue, when we talk to patients. So, what do we know about pharmacotherapy agents and obesity treatment within the context of physical activity? Not much. One study that's big enough to say anything, and they lost about 10 kgs in about an 8 to 12 week period with non-pharmacotherapy agents before they added exercise and before they added pharmacotherapy. So we have no big studies that have been published, well-designed, well-controlled studies that have added exercise to pharmacotherapy for obesity treatment at the onset to make any kind of major conclusions. There's a few that are ongoing, but nothing definitive at this point. So, what then have we learned from other studies of obesity treatment that have used exercise. Well, I'll go back 30 years and talk about some of our very low energy or very low calorie diet studies that were getting weight losses, of big magnitudes that are very similar to what we're seeing with these pharmacotherapy agents. And we added exercise to those treatments. In fact, we did a variety of studies back in the day where we had these very low energy diets. They were about 500 to 800 calories intake per day. They were those diets with endurance training or what some might call aerobic training. We had those same diets with resistance exercise training or strength training, and then we had the same diet with a combination. And what did we find in those studies a long time ago that might inform where we are today? Well, here are the endurance or aerobic training paradigms. These are all supervised exercise sessions in our laboratory, so we know that they did this. And you can see how we ramped them up. This was not a low-intensity intervention. We were pushing them hard, and we supervised every minute of their exercise. The resistance training was high-intensity resistance training. This was going to build strength. This was going to do it. And we supervised every rep and set that these people did. So what did we find? Even though this may look like it's different, there was no significant difference between the groups when it came to the weight that we observed, whether or not we added exercise or not. Looks like there might be, but not statistically significant, and it's a kg more. So what about change in fat-free mass? At the time, that was we used techniques that measured fat-free mass. No significant difference with the addition of exercise to these low-energy diets. It didn't help with the fat mass loss. It didn't help with the lean mass loss. It looks like it might have preserved it a little bit, but no. Look here. These are not statistically different. The exercise did not preserve the loss of lean mass with weight loss. Remember, these are not the same, and it's maybe not volume, maybe it's quality. So what else did we find? Well, we did strength testing on them. We created a strength index by having them do a variety of exercises, and darn it, even though people were losing lean tissue, lean mass, that was equal to what they lost with the diet alone, the people that had a resistance training program added to their intervention got stronger. Even though they didn't preserve the lean mass, they got stronger. The people that had endurance or aerobic training got more fit from a cardiovascular perspective, even though their lean tissue was going down. Those are indicators of tissue quality improving, even though we had a loss of tissue. That's important for us to remember, that it's about the quality. And if you have individuals who are losing weight and losing some lean mass or some potentially muscle, that what remains is what matters. And how good that is important. What about bariatric surgery? We did a supervised study of 6 months of exercise following bariatric surgery compared to bariatric surgery alone. All these were Roux-en-Y gastric bypass patients. Body composition changes after those 6 months was not different between the two conditions. However, we found that the group that exercised compared to those who didn't had improved cardiorespiratory fitness. We found that those who exercised compared to those who didn't had greater improvements in insulin sensitivity. We wouldn't have detected that by measuring body composition. We needed to measure it in other ways. And so I think we need to be thinking about what else is improving with exercise beyond getting fixated on some things that may be not the best measures. And so just going back to this, even though we're seeing this loss of lean tissue, fitness will improve most likely. And then the few small studies that have been done, fitness will improve even though this is occurring. And markers of fitness, insulin resistance, cardiorespiratory fitness, strength, whatever they are, will improve. So the one thing, though, about these medications, and I think that you all face in a clinical environment, is exercise is recommended, But when we've surveyed our patients who have been prescribed these medications, these GLP-1 based medications, okay, most people are not exercising. And most people are not exercising enough. And you can see very few on their own are doing resistance training or other forms of exercise that are going to make a big difference. We also have found that some people report weakness and fatigue in response to these medications, and those are the same individuals who are also reporting low levels of physical activity. We don't have the direction of this. It could be that they're less active, so they're feeling more weakness and fatigue, or it could be these medications are causing some weakness and fatigue that are hurting their ability to go out and be more physically active. And we have to think about then how do we position activity within this context. And my colleague Renee Rogers published this paper. It should be available, I think, in the Health and Fitness Journal of the American College of Sports Medicine that helps to position exercise within the context of these anti-obesity medications from a practical clinical perspective. And as Carol mentioned, just a few months ago, we published an update to the American College of Sports Medicine consensus paper on where physical activity fits within the context of obesity treatment and obesity care. This paper is available on the American College of Sports Medicine website as a free download. It's acsm.org. You can get it there. And in that paper, we provide very specific guidelines and recommendations for physical activity where we say include both forms of physical activity, and that it's inclusion of physical activity, and we have to think beyond physical activity for weight and body composition change and think about it more from a holistic health and well-being perspective, and also to think about things like kinesthetic awareness, fall prevention, and those types of things. The other thing I just want to point out, and you can go in there and get this, is this quick figure that I think is really important. This figure is in there, and it shows you, if I can, when you do things that are going to influence the energy intake or restrict energy intake, basically it's a hypothetical, theoretical model of what it will do. But I want you to pay attention to the light blue colored things. The light blue colored things are physical activity, different forms of physical activity, and the light blue things are the things that physical activity will impact that changes in dietary intake and energy restriction will likely not. And there's a lot of light blue on that figure when it comes to treating patients with obesity. So activity has to be a part of this. So within the couple of minutes that I have left, I just want to point out a couple of things to you that I think are very, very important in this context. And I'm sure that there'll be hopefully some questions. Obesity, we realize, is a disease, right? And we have to understand that those causes and contributors are going to vary from person to person on different treatments and so on. And I think we've come to that realization. I want to keep going back to, lean mass is not muscle mass, mass is not quality, and to get you thinking about quality of tissue when it comes to exercise. We have to think about treatment for obesity, not treatment for weight loss. Weight is one component of obesity, but when we think about obesity as a chronic condition, it's all the other downstream factors. And how do we go beyond treating weight to treating obesity? And if we're treating obesity, activity is going to affect some of those downstream factors differently or in an enhanced way compared to just thinking about it from a weight perspective. And I think that's critical. And the last thing is that we need to make sure that we're thinking about holistic health and well-being. And just like I would not stand here and tell you that I know how to treat cancer, I also hope that you recognize that physical activity and exercise is not telling someone to go and do it. We realize that there's a lot of barriers that people face and a lot of different things that people have. And we should be thinking about the experts in physical activity. And I would encourage all of your clinical care teams to employ a clinical certified exercise physiologist as part of that. And that is different than a physical therapist. They're both important. But I encourage you to do that if you're really interested in the overall health and well-being of your patients. And so with that, I will end basically with this. I'm here to tell you that exercise is critically important, but I want you to think about exercise beyond weight. I want you to think about exercise and physical activity as more in a holistic way. I want you to also think about it and get away from it being a method-centered approach. Resistance training is good, endurance training is good, aerobics training is good. And think about it from a patient-centered perspective, just like you would in any other treatment. And that's going to vary from person to person. And if we do that, our opportunities are huge because we know that most patients with obesity are not active enough. And we have many benefits from a health perspective that occur whether or not we move the scale when it comes to physical activity. So with that, I will thank you for your time. Mm-hmm. Do you want me there or here? I'll go there.
Okay, lots and lots of questions, and we don't have much time. A couple of questions, John. Does exercise reduce visceral fat? And does exercise improve the metabolic rate? And if so, what kind?
Two quick questions. So, does it improve visceral adiposity? There have been plenty of studies now that have shown that even without weight loss on the scale, exercise targets visceral adiposity in a big way. So visceral adiposity is a main target of most forms of physical activity with enough intensity, and we see it both with resistance exercise as well as sufficient endurance or aerobic types of exercise. Does exercise increase metabolic rate? No, it does not. Plenty of studies that have shown this. When you look cross-sectionally at the data of active people versus less, many active people have a higher metabolic rate. But in training studies, where you train people and do supervised training and you measure metabolic rate, it barely moves. And the driver of that is probably muscle mass, but it takes a ton of exercise over a long period to do that. So it may suppress a loss of metabolic rate, but it really doesn't improve resting metabolic rate very much at all.
Thank you. Microphone.
Nick Murray from Adelaide, Australia. I've got a question primarily for Dr. Jakicic, and it's about language and communication. About 20 or 30% of young women in most Western societies have an eating disorder. You could not give the talk you just gave in the way you gave it to an eating disorder meeting, which is not a criticism of the talk you've given. How do we reconcile approaching these two important health problems?
Yeah, well, it's a very good question. I'm speaking from a context of individuals who have presented for obesity treatment. And when we do that, we are actually finding that very few who present for treatment at least have an anorexia type of concern. Some of them have binge eating disorder or tendencies for binge eating, but they aren't presenting for our weight loss programs. But you're correct, we have to be very cautious about this. And that's why I think it goes back to this individualizing the prescription as opposed to it's a one-size-fits-all. I think we want to be very, very careful about that. But I can also say that in our weight loss programs, we are not inducing eating disorders from that perspective. But you're correct, there are people we have to be very cautious about, and that's where the caution has to come.
A couple of other questions that have come in, and then we'll get to your question. Dr. Duggan, several people want to know if there is epigenetic reversal with weight training. And while you're at it, they would also like to know, does caloric restriction affect epigenetic changes in offspring? In other words, does caloric restriction in the mother affect epigenetic changes in the offspring? And from your talk, I would guess that they do.
Yes, it does. It depends on the degree of caloric restriction, but from the data from children of women who were pregnant during the Dutch hunger winter, that there is metabolic, there's epigenetic changes to genes in IGF, for example, that persist 2 generations. And some of these epigenetic inheritance affects many metabolic pathways leading to obesity, for example. And the first one about, yes, there are epigenetic changes with exercise. They tend to be obviously localized to muscle tissue and impact sort of increases in IL-6 production during acute bouts of exercise.
But I would guess if exercise affects visceral adiposity, you would see systemic changes as well.
Yeah, the issue is a lot of the epigenetic changes are very localized, but some of the microRNAs are excreted into circulation. So it's trying to, work out what's very localized, what affects local tissues and systemic changes as well.
Thank you. The center microphone, please identify yourself.
Yeah, I'm Ishmael Jatoy from UT San Antonio. I was wondering if you'd comment on the qualitative age interaction that's oftentimes evident in studies that looked at the association between obesity and breast cancer risk. So obesity seems to increase breast cancer risk in postmenopausal women, but seems to have the opposite effect in premenopausal women, seems to decrease risk. Do you have any thoughts about that?
I don't know who wants to tackle that. I guess we can both tackle that. You want to start off? Well, I think the issue with very young premenopausal women probably relates to, in the very obese women, they may have anovulatory periods, they may have reduction in progesterone, and a lot of other things. There is some indication, however, that particularly for BRCA1 mutation carriers and or triple-negative breast cancers, that premenopausal obesity may play a role in breast cancer. Neil, you want to comment?
Yeah, I agree. I mean, I think also it has to do with
your microphone's
can
not
folks
working
hear
very
me?
well up here.
Yes? Okay, we're good? All right, great. So yes, I think, and this has been looked at a number of different ways. I don't think we have a definitive answer yet, but I think some of the key questions are the duration of obesity and the onset of obesity in the pre versus postmenopausal setting. The biology of the breast cancer itself, we know, is of course different, obviously in the premenopausal setting versus the postmenopausal setting, where growth factors may be less hormone dependent in the premenopausal setting. I think it's less, we have evidence to suggest that it's less of a detection issue as once thought. And then the last thing I think is important is the adiposity question. Much of the data with regard to BMI and breast cancer risk in the pre- and postmenopausal setting is really based on anthropometric assessment of the individual rather than body composition, but more importantly, the quality of the microenvironment, getting to your concept, the quality of the adipose tissue.
I want to get to a couple of quick issues regarding the Incretin mimetics that we are very frequently asked. Number one, since about a third of the patients in my clinic, I don't know about anybody else's, show up on these things.
To Dr. Duggan, Sebastian Anton from University of Calabria, about the frequent methylation event occurring in the obese patients. Have you looked at the methylation of SRA1 gene? I mean, the gene codifying for estrogen receptor alpha, because this may explain, in some circumstances, the onset of endocrine resistance.
Catherine, I think that he's asking a question about, have you looked at methylation of the ESR gene?
No, not in our lab, but there is evidence to suggest that obesity, there is alternative methylation patterns and downstream estrogen receptor signaling pathways, depending on whether or not the patient is overweight or not. And that SERMs, used widely in chemoprevention and treatment, alter methylation pathways in breast. Though given its tissue specificities, it's not clear in other tissues what happens there. I hope that answered your question.
Yeah. We only have 1 minute left before we have to vacate the room. So everybody that has more questions, please get with the moderators and the speakers at the end of the session. But one thing that keeps coming up over and over again is that so many people that are on endocrine agents are also now starting to take the incretin agonists. And what do we know about drug interactions or change in endocrine agent absorption in women using the FDA-approved drugs and the plethora of the compounded agents that are being prescribed online for these patients? Neil, do you have any thoughts about that?
This is a critical question, and the short answer is we don't know anything. And I think the big issue here is that for patients that are on curative intent therapy, are we potentially doing them a disservice? I think that the key question in my mind is that the anti-adiposity effects or the pro-metabolic effects are likely to outweigh concerns about drug absorption with regard to their endocrine therapy, for example. We know that gastric slowing is involved here. If anything, it might theoretically exacerbate the adverse effects from endocrine therapy. But on the flip side, we also know that the weight loss and the loss of adiposity likely is going to improve the arthralgias and many other adverse effects associated with endocrine therapy in the post-diagnosis setting. So we certainly need prospective randomized control trials in the post-diagnosis setting to address those questions. But I suspect that the pro-metabolic effects will likely, on balance, be beneficial.
And I would like to close for the researchers in the room a plea, particularly for Lilly and other companies that are involved in making these agents. There is such a dearth of trials, either in breast cancer prevention or breast cancer survivorship with these agents, mainly because of lack of supply of the drug. We desperately need more information on how these agents are affecting our patients. And I thank you all so much for attending and staying through the whole session. Thank you.